So we just finished a video looking at the structure of nucleotides and how we can hook nucleotides together with a phosphodiester linkage to make nucleic acids. And we had the generic structure that we started with for a dinucleotide, and then we learned how to map on specific information from an example structure that I might ask you to draw. So what we're going to move on to now is, remember, we talked about how you can have a very special nucleotide, ATP, that can serve as an energy source. So we're going to look at the structure of ATP. We're going to draw it and then understand why that structure sort of affords its function. And we're going to do it specifically for, again, that biological money that is ATP. So highlighting, right, I want you to remember that ATP is your biological money, right? And when we think about the structure, we're going to be able to understand why. So we're going to rely on some of the structural information that we learned about when we drew our nucleic acids, right? We have a ribose sugar, and again, ATP is a ribose flavor, so it's going to have at that 2' position, it's going to have a hydroxyl group. Now in this case, I actually want to draw out what the base is for adenine. So it's a purine nucleotide, so it's going to have a 5-membered ring here, and then it has a 6-membered ring, and that 6-membered ring has 2 nitrogens in it, and I like to call this the flagship nucleotide because it has the only exocyclic sort of functional group it has there is a primary amine. Okay, so this is the structure, and if we stopped here, again, we have to have this group here to indicate we'd have a hydroxyl group there. If I stopped here and I just drew my sugar and my base, this would be a nucleoside. But remember, we're drawing a nucleotide, and so I'm going to have, in this case, 3 phosphates that are with it. So I'm going to draw all of my phosphates out here. Hopefully I have enough room here. I've got a phosphate, phosphorus, oxygen, phosphorus, oxygen, phosphorus, oxygen. I got all of them in. All right, going to put in all of my negative charges there, and as you're going to see in a minute, that's really important. Okay, so in this case, highlighting adenosine triphosphate is important because I have 3 phosphates here. So 3 phosphorus atoms. So again, this is a triphosphate. So again, that triphosphate comes here. I'm going to move this down a little bit because I want to make sure to highlight there is some formalism for how we sort of think about these phosphates. So again, this is each one I'm sort of bracketing here. We give Greek letters to indicate their position to the nucleobase. So the first one away is referred to as the alpha phosphate. The second one away is indicated as the beta phosphate. And then the terminal or third one away is considered to be the gamma phosphate. So alpha, beta, gamma, those first 3 letters in the alphabet. Some other things to highlight about this structure that make it kind of unique is if we look at what we have here, we have 4 negative charges. So this is a huge polyanionic species, so 4 negative charges. And if you think about whether 4 negative charges would want to be close to one another, they wouldn't. So that's highly unstable, right? Now, when I say highly unstable, remember when we talked about our ATP being valuable but stable, okay? It is stable in this structure, but it really would love to have those negative charges separated, which is why it's valuable. So we've got those 4 negative charges, again, which is a high energy sort of state. Maybe that's a better way to state it. It's not that it's unstable, it's just very high energy. And remember, when we talked about things being high energy, we used swiggle bonds like this. So I've got 2 bonds here that we call high energy or squiggle bonds. Okay, and so these are high energy bonds. Okay, so these bonds are why ATP is worth that magic 30.5 kilojoules per mole. And what we're going to see is when we break either of these bonds, and we'll talk about the different pieces that we get when we do break them, we separate those negative charges, and that helps increase stability in our system. Our system goes downhill in energy, so that excess energy is given off. So that's fundamentally why ATP is worth money, is when we break or separate these high energy bonds. So when we break these high energy bonds, we are going to separate those negative charges, and that makes it lower in energy. And so the excess energy then gets to be given off. So if you remember when we looked at our reaction coordinate and understood why ATP was valuable but stable but accessible, do you remember this? We didn't look at the structure. We just said ATP was valuable because when you hydrolyze it, you get ADP and PI. And we knew that that was valuable. 30.5 kilojoules per mole of energy would be given off. Right? We talked about, though, why it was stable, because there was a very large sort of transition state that you needed to go through, a very large delta G double dagger. So that's why our money is stable. But again, enzymes are what make that money accessible because they lower the energy of the transition state and they allow us to proceed through with a much smaller delta G double dagger. All right, so coming back here, now that we know the structure and we understand when we break these bonds, that's why we get that energy out. But let's sort of talk about what happens when we do this hydrolysis chemistry. When we do this hydrolysis chemistry, if we break bond number one, okay, if we break bond number one out here, okay, we are going to be removing the gamma phosphate. That is going to leave us with ADP and then inorganic phosphate. That's the reaction that we just saw with our reaction coordinate. Delta G in that case is going to be equal to minus 30.5 kilojoules per mole, right? So remember, when we're talking about this, when we talk about the delta G for this reaction, we can say that it's a negative 30.5 kilojoules per mole because we know it's a downhill reaction. If I ask you the question, how much energy is ATP worth? You just say 30.5 kilojoules per mole because we know that energy is given off in this hydrolysis reaction. Okay, so this is the chemistry again that we talked about with our reaction coordinate. So I'm just going to put like a little reminder here. This was our reaction coordinate chemistry. Now, one other reaction that I want to make sure to remind you of is we can also do hydrolysis chemistry where we're going to break that second bond, okay? Not the first bond. So look what happens here. If I break this here, what I'm left with is an adenosine with one phosphate. So I'm left with AMP, and maybe you remember when I have two phosphates that are hooked together, this is called pyrophosphate. So this is going to lead me to AMP and then PPI, which is again pyrophosphate. And why pyrophosphate is important is because you have a lot of the enzyme pyrophosphatase in your body. So pyrophosphatase, and that is going to, in a very important way, let's go ahead and just say, let's imagine that these reactions are reversible. The pyrophosphatase reaction is irreversible. And that's an important piece here. I'm going to put in here that this is irreversible. And what it does is it's going to finish, remember this is pyrophosphate right here. It still has that number one bond that's intact. That's still a high energy bond. When I break that bond, then I'm going to have two inorganic phosphates, and then I'm going to get a bunch of energy out as well. This is a huge downhill reaction, which is what makes it highly irreversible. But here's why this is important. If you have any biochemical process that's reversible and it generates pyrophosphate, the fact that there's tons of pyrophosphatase around in your body means it's going to find any PPI that's around there and it's going to react it and it's going to remove it from solution. Le Chatelier's principle will tell us that when you remove this from solution, you allow this equilibrium to continue to shift forward because it needs to replenish that. So even if this was not a very favorable reaction, any reaction that generates pyrophosphate can be pulled forward by Le Chatelier's principle by the prevalence of pyrophosphate in your body. Okay, so some neat little applications to think about how specifically the structure of ATP is what affords its magic value of 30.5 kilojoules per mole.